All right, our second speaker is Yusuke Himeoka from the University of Tokyo, and he'll tell us about theoretical basis for cell deaths. Please.
So yeah, thank you, Prof, for having me here, and yeah, this great opportunity. But the problem and the David, I'm sorry that you heard exactly the same talk 2 weeks before, maybe. So I'm going to talk about some kind of theoretical approach for understanding cell death. It's not pretty much related to autocatalysis, but I mean the living system is kind of autocatalytic system in the sense the cell death, in the sense the collapsion of the living system is somehow collapsion of the autocatalytic system will be related. And let me briefly introduce myself. I got PhD with the Kunihiko Kaneko as the supervisor, and after that I got to the Copenhagen University. I did 2.5 years postdoc, but the latter half was under very strict COVID restriction, so I couldn't enjoy the Copenhagen that much. But yeah, and I'm now assistant professor in the University of Tokyo in Furusawa Chikara's lab. Here we have doing both. I'm not doing, but our group is doing experiment and the theory, and our professor is very famous, especially in Japan, for laboratory automation technique. So I'm pretty much interested in kind of bacterial physiology and the very stressed condition or brink of death in a sense, and in terms of metabolism. Then I want to kind of answer the question like, what is the inherent difference between living system and the non-living system that we call just chemical reaction system? And also, we human beings have never succeeded to construct cell from scratch. Then I want to understand why it is so hard, or why it is believed to be so hard to construct living system from scratch. So I'm basically working on two types of project. One is the stability of the metabolic reaction system, but today I'm going to talk about the quantification theoretical attempt to quantify or understand life-death boundary. So I mainly focus on the cell death, not the individual death of the microbes, because the unicellular organisms, individual cell and individual death and the cell death are the same thing. And in this decade, we have many interesting single cell level measurement of the cell death. For instance, this is yeast, this is E. coli. There are trade-off between growth and death, and it is very kind of carefully measured what happened under the starvation condition and blah, 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 what is related to the probability of regrowth after the starvation or something like that. But I'm very happy of kind of seeing those experimental results, but I'm a little bit frustrated because I mean, yeah. Kind of frustrated because the definition of death is totally different among the papers. And also, yeah, first of all, it's not pretty much defined. So what I want to do is kind of try to define death in a mathematical sense. Of course, in experimental approach, they have some experimental assays that is called cell viability assays or cell death assays. Suppose that the situation that you want to judge whether your cell after starvation is living or not, then the very direct way of measuring it, judging it, is to kind of plating them or see whether they start to grow if you give them fresh nutrient under the microscope or not. Then this is one of the such method, but this is a bit annoying because labor intensive, because you have to dilute the cell culture, and then you have to plate it, you have to put it to the incubator, wait for a while, then you have to count the colony. Then there are some shortcuts like PI staining, which is basically measuring the damage on the membrane. And the logic behind is that if your cell has very damaged membrane, it is very difficult to regrow. And also this metabolic activity kind of measurement called MTT assay, measures specific reaction, converting the yellow fluorescent chemical into purple fluorescent chemical. So basically, if your cell is still kind of capable to perform this reaction, you can see the color change from yellow to purple. But there are some contradiction among the different techniques. So here you see in this paper, they kind of tried to kill the E. coli cell and the Staphylococcus cells using 16 different stressors. Then they quantified the cell viability using plating assay and the MTT conversion assay. And the y-axis is, I think it's log, but I don't think they put any description on the paper. That's a bit weird, though. So basically, what is this figure telling is that if you see the blue one, this is E. coli result. If you expose the E. coli to the hydrogen peroxide, then you kill cell a lot because the maybe log of CFU enumeration outcome is close to -10. It decreases a lot. But if you measure the outcome of the hydrogen peroxide exposure using the MTT conversion, then the effect is almost zero. On the other hand, if you expose the cell to the silver, it does nothing, basically nothing in terms of the CFU, but it decreases a lot the MTT conversion efficiency. So which technique you use, the outcome or your cell death judgment would be totally different. So I feel that we are at the stage that seriously consider what the death should be. But of course, this is very kind of philosophical question. So what I want to do is just kind of play around with some concrete metabolic model, then try to understand something out of it. So the kinetic model that I play around is this. This is not my model, proposed by the German group. Of the equals metabolism under anaerobic condition. And this is central carbon metabolism. Glucose is taken from extracellular environment, and it is metabolized through the glycolysis. And this is fermentation pathway, and it has some TCA cycle. But this is anaerobic condition, so TCA cycle is incomplete. This is basically used just for making the precursor of amino acid. And here I also draw some substrate level regulation. Blue is feedback inhibition, not necessarily feedback, but the substrate level inhibition, and the red arrow is the activation. And there are some metabolites coupled to the reaction. So red one is ATP, pale red is ADP, this is NADH, this is NAD or something like that. And the model is implemented by using the ordinary differential equation, where X is the concentration of metabolite, S is stoichiometric matrix, and the V is the reaction rate function. This is function of the concentration X. And example is something like this. This is very complicated, but you don't have to remember it. Only the thing that I want to emphasize is that in this type of the model, concentration of enzymes are considered to be constant. So this is delta's parameter. And also, this model cell has a biomass formation reaction with which cell is considered to kind of perform volume growth. Oops. And because this is ordinary differential equation model, it has attractors. And on the attractor where a cell is growing, the reaction flux flows something like this. So yeah, this looks very healthy, right? From extracellular environment, glucose is taken up and it is metabolized. And ATP is produced, and NADH is produced, and so on. But interestingly, if you kind of perturb or choose different initial condition of the metabolite concentration, it shows it goes to the other attractor. As far as I confirmed, this model has bistability. I didn't see 3 or more, but yeah. So this is the time course of the this ordinary differential equation model. In different color indicates the concentration of different metabolite And then the x and the y-axis is in logarithmic scale. So in a growing state, which is this. So here in this on this attractor, the concentration of metabolite is very kind of reasonable, experimental reasonable range. And also, while I'm not showing it, the growth rate at this attractor is kind of doubling time as 2 hours or something like that. This is consistent rate of with the experiment. Then, but if you change the initial condition, it goes to the other attractor where the it shows some kind of anomalous relaxation, power law relaxation, then reaches to the kind of some weird attractor where the concentration of metabolite is pretty low, and the growth rate is also, this is not showing here, very low, almost 0. And the flux distribution at this attractor is something like this. So glucose is taken up from the extracellular environment, and almost all the glucose taken up is secreted as lactate. So actually, this type of metabolic flux is experimentally confirmed using lactobacillus. So I think this is kind of capturing reality. So the reason why I'm working on kind of playing around with this metabolic model is that I expect I can learn something about this. So I want to think about whether it is reasonable to term this non-growing state as the dead state, because I mean, it's presumably dead. I don't know. So then the question is this. So can we term the non-growing state as a dead state naively because it is not growing? My answer is we shouldn't, because there are 2 concerns. One is from dynamical system point of view. As I told you, in this model, the concentration of enzymes are fixed constant, so it's a parameter. Then if I kind of see that non-growing state as a dead state, then the take-home message would be the death is being kicked out from the basing of the growing state. I don't believe we can learn anything from it. And the second concern is more from the biological viewpoint. Again, as I said, the enzyme concentration is constant. So if this state is dead state, we are basically admitting that the cell is not doing anything while their intracellular metabolic concentration is getting weird. So basically what is lacking is the next question. So I think the lesson from the plant seed would be very helpful to think about how we should define death. So the question is, do we consider plant seeds that are apparently inactive, so it's not growing, to be dead? We don't call it dead body, because we know that there's some operation that kind of resume the activity, right? Which is kind of putting seed onto some soil and give some water and etc. So what was lacking on the previous argument was probably there's the viewpoint of the control. So then our proposal of definition of cell death is the following. So suppose that we have one, not necessarily one of few point that is the reference of the living state that we call representative living state. Then if you want to judge your given state X is living or not, then you should check if there's control from your state X to the reference state of living, representative living state, by modulating the enzyme concentration, which might be controlled by the gene expression level by cells themselves, right? And also the external concentration of metabolite, which is something we can control. If there's no control to bring the system X back to the referential state of living, then we call this state X as dead state. So schematically, this is something like this. So here's the trajectory, uncontrolled trajectory from the growing state to the non-growing state. Then if you take the concentration from very beginning of this relaxation, then if you start to control the enzymatic concentration, then you can control the system back. But if you're taking the concentration from very later stage of the relaxation, then you cannot. Then it will be reasonable to call this final state as a dead state, and also there's point of no return somewhere between this point and this point. This is the basically idea. Then to answer to the following this kind of definition of death, to check whether a given state is dead or not, we have to be able to compute the controllability problem. So our ordinary differential equation is something like this. X is concentration metabolite, and U is now control, which represents the concentration of enzymes. And here, O dot is Adamov product, element-wise product of the vector. The V is a nonlinear, the reaction rate function. So this type of controllability problem is quite difficult to solve because X represents the living state is global control, and the U is the concentration of enzymes, which must be non-negative, and the V is a nonlinear function of X. So those three difficulties summed up, and we have the restricted global nonlinear controllability problem. So of course, there are some method to compute such kind of controllability problem using especially Hamilton-Jacobi-Bellman equation. But using those equation, it is very difficult to tell, to kind of prove the absence of the control. So if you could find the control, maybe you are basically lucky. But if you cannot find any control, bring the system back to your target state, it is not telling you that there's no control. So there's a possibility that you just had a bad luck and you couldn't find it. So we developed a simple mathematical framework to compute the controllability for the catalytic reaction system that we call stoichiometric rays. So this is a bit mathematically technical. So I just give you some intuition of this controllability method. So our method, stoichiometric rays, utilizes the very important feature of the catalytic reactions, which is that catalysts do not change the equilibrium. It just changes the activation energy. To mathematically rephrase this feature, we suppose the reaction rate function has this decomposition. So V is a reaction rate function. This is decomposed into the part we call kinetic part and the thermodynamic part. Kinetic part is just a strictly positive function of X, and P is this type of kind of mass action type of kinetics. And I think that almost all the popular the biochemical reaction rate function has this decomposition. For instance, in a reversible mechanism engine, this numerator is P, and the remaining parts are F. So this is strictly positive, and the P, the numerator has this form. And then once this decomposition is accepted, then our ODE is something like this. Then important point is that U is positive, non-negative, and F is positive, and the P is only the part that can take positive and the negative. And also X is not depend on U. It is pure function of X. This is rephrasing of the catalyst does not change equilibrium, because even if you change amount of enzymes, you cannot directly change the direction of the reaction. If you want to change the direction of the reaction, you have to control the metabolite concentration. So indirectly, of course, you can change the direction, but you cannot directly change the direction. This is rephrased. So by using it, we can actually compute the controllability of the catalytic reaction system. This is just a toy example. We use the reversible bracellator. It has 3 reactions and the 2 metabolites, two variables. And here I draw the vector field by changing the control parameter just kind of sinusoidally. And then what we want to do is that checking whether it is possible to control the system from this x0 to x star. Then it's presumably not that difficult, but let me just try. So to check the controllability, we introduced the balance manifold and the direction subset. Balance manifold is 0 set of each thermodynamic part function. So on it's kind of subset on which the reaction are equilibrate. Then I think the first reaction, balanced manifold of the first reaction is easy to describe. The first reaction is secretion and the uptake reaction of chemical A. So if A becomes 1 over K0, it stops. So the balanced manifold of the first reaction is this one, right? And then by the direction subset, the four-phase field is partitioned onto several subset. And the important point is that on each direction subset, the direction of the reaction is set to the fixed direction. So which means that the here red arrow is the stoichiometric vector of the reaction, first reaction. It just kind of increase or decrease chemical A. And in the left side of this balanced manifold, the stoichiometric vector of the reaction 1 is pointing to the right because it just increases A. And in the right side, it decreases A, so it is pointing to the left. And the same applied for the stoichiometric vector of other two reactions. And then the most important part is this, because this reaction, this ordinary differential equation is generated only those three reactions. So we can say that the all the vector field realized in this model is should be generated by the non-negative linear combination of the stoichiometric vector put in each direction subset. And also the converse also holds. If you are able to generate a certain kind of state transition by combining stoichiometric vector in each direction, non-negative linear combination of stoichiometric vector, then you can actually generate such transition. So which means that from this x0, you can reach this red field region. So the upper extreme ray is kind of generated by just using only red reaction. And this kind of bottom extreme ray is generated by either using cyan or green one. So by doing that, you can fill the reachable region. Then from this, once you reach this balanced manifold, you can somehow enter the other direction subset. And in this direction subset, the direction of the stoichiometric vector of the first reaction flipped to the left. Then by adding this red vector and either green or cyan, you can make the kind of straight down vector. Then this kind of field region is where you can go. And from there, you can enter this direction subset. From here, you can enter this direction subset. So basically, you can control the system from x0 to x star. And interesting thing is that if you are if your target point is here, you are not kind of reachable, controllable from x0 to x star. So if you choose the representative living state to be here, this x0 is living state. But if you choose x star slightly below, you are not living. This is basically the framework, and there is some math, but I skipped this. So then what I want to do is applying this method to kind of check the point of no return and the final state is dead or not in this model. So here I, for instance, sample the concentration from this time point. Then from this time point, I start the control and check whether it is possible to bring this concentration to the growing attractor. Then if you see any kind of red color, it indicates that there's control back to the system. So here it's revivable, it is living, but all of a sudden, you are no longer living, no longer controllable to the growing state. So after that, you see all black. So what this figure is telling is that actually the final state is the dead state in terms of the kind of loss of the controllability to the growing state. And also here's the point of no return of this trajectory. Then, so I'm just kind of, as a demonstration, I show the controllability computation for a single trajectory, but of course we can do the same thing for a number of different trajectories. Then schematically, we have something like this. So for all the trajectory going to the dead state, we have point of no return for the single point of no return for each trajectory. Then connecting those, we can separate the phase space into the living state and the dead state. So then this surface separating the living region and the dead region that we call the separating alive and the non-life zone. And as an abbreviation, we call this a Sanzu hypersurface. And the Sanzu is named after the Japanese Buddhism tradition, the Sanzu River. This is a mythical river that separates the world of life and afterlife. So of course, this is not just conceptual thing. We can compute which metabolite is important for kind of setting the Sanzu hypersurface of this model. So I skipped the details, but acetaldehyde, ethanol, and the NADH are kind of turned to be very important. And to check it, I'm now setting up the single cell level experiment with my experimental collaborator. So basically, this is it. Our proposal is to define that state as the loss of controllability of the representative living state. So I think I didn't explain in detail about the representative living state, but maybe I don't know whether you know that there is some controllability cellular field called as viability theory. In the viability theory, you are required to first draw the boundary of living state and the non-living state by yourself. So basically, you are required to list all the living state. Then viability theory tells you which type of control kind of prevent your kind of system goes outside of the living region. But in our approach is basically you are required to list only a single or a few representative state of living that you can for sure say, okay, this state is living. Then based on the controllability, this framework automatically draw the boundary between living and non-living. So if you have to list all the living state, you have to judge by yourself whether dormant state or spore state are living or not. But in our framework, basically the dormant state, the spore state can return to the growing state. Then based on the controllability, you can maybe automatically see the dormant state, spore state is living. So this is a big difference. But there are several kind of incompleteness of the theory. For instance, that we are implicitly assuming that the perfect observation of intracellular state, and also we didn't add any cost on the controller, blah, blah, blah. But yeah, so I think this is kind of one of the first step to understand what is death in theoretical manner. So thank you very much. This is it.
Thank you.